Hi, it's Professor Schmidt, and let's solve some problems about meiosis, gametogenesis, and sexual life cycles. So in question 1, it asks us, a particular animal has a diploid chromosome number of 50. At metaphase 2 in meiosis, how many chromatids are present? So while they're specifically asking about metaphase and meiosis here, it might be instructive to just go through a couple of aspects of both mitosis and meiosis real quick, just to see how many chromatids would be present in various times, and then we could just make a generalization. They're asking here about some animal with 50 chromosomes, but humans have 46. Whatever you encounter, you should have this general idea in your mind. So let's just say we were talking about mitosis, If we were talking about mitosis, prior to S phase, say this animal cell was in G1, there would be 50 chromosomes, right? And since they have not been doubled yet, there would also be a grand total of 50 chromatids. Because those type of chromosomes, it sounds a bit awkward, but the definition of them is that one chromosome is composed of one chromatid. After S phase, we would still say there are 50 chromosomes, but they've been doubled, right? So now there are 100 total chromatids. Now remember, I said we were dealing with mitosis, but what I really should have said is that this starting cell, whether it's going to undergo mitosis or meiosis, this initial situation is similar, right? This cell, whether it's going to go to mitosis or meiosis, it starts off with 50 chromosomes and 50 chromatids, and after S phase, there are 50 chromosomes, each of which is composed of 2 chromatids for a grand total of 100. Now, depending on which cell division process we're going to go through, that's what's going to be able to answer our question. If it was mitosis, right, there would be 100 chromatids in that cell until mitosis was complete, and basically remember what we're doing is separating the sister chromatids, half of them going to one cell and half going to the other, so that when all was said and done, there would be 50 chromatids in each of the new cells and also 50 chromosomes, because now again we're back to those chromosomes being composed of one chromatid. But in the case of meiosis, let's see what's going on. So if it was meiosis, we're still starting with a cell that has 100 chromatids, right? Now, remember what happens in meiosis 1. They're just looking for a number here, but we want to remind ourselves that homologous pairs are lined up. So there's 2 homologous chromosomes. There's a total of 4 chromatids there. Meiosis 1 separates them like this. So even though that's important to know to understand meiosis 1, the bottom line with the numbers is that the 2 new cells after meiosis 1, right, we're going to have one cell that has, from our drawing, that, and the other one has a similar thing, but considering there are 50 chromosomes and 100 chromatids here, there would be 50 chromatids total in this cell. They're arranged in a particular way. Alright, so after meiosis 1, there would be 50 chromatids in these cells. Let's go back up, and I almost forget what the question was asking. So they're saying now at metaphase 2 in meiosis. So let's just keep track of one of these cells. It's going to go into the second division, right? At metaphase 2, I'm not going to draw all of them, but all of these are going to be lined up, and the sister chromatids have not been taken apart yet. So the answer is at metaphase 2, there is going to be 50 chromatids, and that should be our answer. So that is the answer. We could be done right now, but I just want to mention that if you took that cell and allowed it to complete meiosis II, right? It would be totally done. These new cells would be gametes or some kind of pregametes, and they would have 25 chromatids in them and 25 chromosomes as well, right? Because remember, the whole point of meiosis is to reduce the chromosome number. So we started with 50 chromosomes in this organism, and their gametes, which would be after meiosis II, would have 25. Now, and they'd be just in this situation. So I went a little overboard in terms of what the question actually asked, but I think if you understand this type of analysis, you could answer many similar questions without having to do it over and over again. So 50 chromatids. Question 2 asks, assume the amount of DNA found in a somatic cell when it is in G1 is represented by the quantity Y. How much DNA is found in each daughter cell after meiosis I, after meiosis II? Now, if you did question 1, this one is rather similar. It's not asking for a number of chromatids, but it's asking basically the same idea. How much DNA total is here? It's more of a generalization because it's basically saying we don't care how many specific chromosomes there are. However many there are, we can apply this. So if you take a cell that's about to go into mitosis or meiosis, right, and say that, okay, it has the quantity Y, remember that means that if it's in G1, let's just say for example, and it didn't go through S phase yet, the chromosomes have not been doubled. As it goes through S phase, each of the chromosomes get doubled, so I hope it makes sense that there'll be double the amount. So by the time you get to G2, there will be 2Y total amount of DNA in there. Whatever Y is, it really doesn't matter. Now, if we were talking about mitosis, right, isn't the whole point of mitosis to make cells that were equal to the original one here? So mitosis pulls apart the sister chromatids, and that means that after mitosis, we would go back to having Y again, right? Just if you're having trouble thinking of this abstract Y number, just assign any number to it. Say it was in humans. You had 46 chromosomes, right? After S phase, chromatid-wise, you'd have 92, and then after mitosis, you'd be back to 46 chromatids and chromosomes, right? Because each chromosome is composed of one chromatid after mitosis. This particular question is asking us about meiosis, right? So, as we said before, just from the point of view of one homologous pair, after meiosis I, we're going to have separated each member of the homologous pair into a new cell. So that means we've cut the total amount of DNA in half. So after meiosis I, we would have, interestingly, Y again, right? Now, if you're assigning the numbers, think of what this means. If we use the human, right? After mitosis, we would have 46 chromosomes, but each of them is made of one chromatid, right? The arrangement's different after meiosis I, but the amount is the same because after meiosis I, we've already done the reduction division. So we have 23 chromosomes, but each of them is made up of 2 chromatids. So if you looked at the total number of chromatids, they'd be 46 in either of these cases. I know even though the question didn't ask about a number of chromatids, I want you to relate it to that. Maybe if you see a question like this again, you won't have to go through all the reasoning, but they're not asking about the arrangement, but I think it helps you to understand that. So after meiosis I, you still have Y. Now from the point of view of one cell, in meiosis II, the sister chromatids are being separated apart, and we're going to make 2 new cells, each of which has one chromosome, well, one member of the one chromatid, which is now one chromosome, right, from that doubled chromosome above. So the bottom line is, if there's Y total amount of DNA after meiosis I, after meiosis II, you would only have 1/2 y in each of those new cells. And that makes sense, right? Because again, if we're assigning the numbers, y equaled 46 in humans, and we know that the gametes, which are what basically form after meiosis II, are going to have 23, which is 1/2 y, 1/2 of 46. So they asked us after meiosis I, the answer is definitely y for that part, and after meiosis II, the answer is definitely 1/2 y. And just to finish it up, we had said that after meiosis I, there were 23 chromosomes if this was a human, each made of 2 chromatids. After meiosis II, there are 23 chromosomes, but each now is made up of only 1 chromatid. So you take half after meiosis I and another half after meiosis II. So y, you may be wondering why. Sorry. Y and 1/2 y after meiosis II. That's the ticket on this one. In question 3, it says the following cells are from an organism with a haploid number of 2. Determine which stage of cell division each cell is in. Now what they want to get at here is you have to determine if meiosis or mitosis is going on, and once you do that, then you need to figure out what stage. But to set the stage, let's just make sure we understand something. They're telling us, the information they're giving us is the haploid number is 2, right? N equals 2. We might as well say that the diploid number is 4, because that'll probably help us to understand that. Another thing that will be vital in understanding this to remember is that the only time doubled homologous pairs are separated from each other or are associating with each other at all is during meiosis, and specifically meiosis I. So keep that in mind. In diagram A over here, we clearly see sister chromatids being pulled apart from one another, which indicates an anaphase of some type. But what we need to ask is, once this anaphase is done, we're going to have 2 new cells, right? And each of them are going to have 2 chromosomes / chromatids in them. If the haploid number is 4 and we're ending up making cells that are going to be 2, we know that that has to be meiosis, and in fact we know that it has to be meiosis II. So A over here is going to be an anaphase of meiosis II, just about finishing things up towards making gametes, right? Now cell B here, remember what I said before, you can tell we wouldn't have known necessarily, except they're different shades of blue, that those were homologous pairs, but now homologous pairs are separating. When does that happen? Well, that happens, again, it's anaphase because it's a separation, but it's anaphase of meiosis II, sorry, of meiosis I. 1 for sure. Let's make sure the numbers make sense, because after meiosis I, we should have a haploid number of chromosomes, so we'll make these cells that are going to come about again. We do. There's 2 total chromosomes in there, but they're still doubled, so there's a total of 4 chromatids. So that's why that's anaphase of meiosis I. Now here, they're lined up in the middle, and that represents a metaphase. So is it meiosis or is it mitosis? Well, again, the sister chromatids are lining up next to each other, and really C is related to B, at least in our mind. C actually occurred right before B, right? Metaphase happens before anaphase. Since the sister chromatids are lining up, it has to be meiosis. And even if you didn't have that other diagram, you could tell this is meiosis I, because that's the only time homologous pairs are separated. In meiosis II, sister chromatids are separated. So this is metaphase of meiosis I. And lastly, in D, there's a total of 4 chromosomes, which is the diploid number, all lined up right in the middle. So lining up in the middle is metaphase, but this has to be metaphase of mitosis, right? Just look at the pattern, different pattern between this metaphase of mitosis and this metaphase of meiosis I. You have all the same chromosomes, but they're lined up in a different way. In mitosis, they line up without any regard to the homologous pairs, whereas in meiosis, the homologous pairs are key. So we have our answers, and that is A is anaphase of meiosis 2, B is anaphase of meiosis 1, C is metaphase of meiosis 1, and D is metaphase of mitosis. Question 4 asks, somatic cells of dolphins, diploid organisms, contain 44 chromosomes. Determine the number of chromosomes and chromatids present at each of the following phases of cell division. Now there are quite a bunch of parts of this question. There's A, B, C, D, E, F, G, H, I, J, even though you can't see them all here. We broke them up so we could consider them a few at a time. But before we even start a problem like this, I think we should, instead of focusing on the specifics of what they're asking, we should get the general idea firstly of what the difference is between a chromosome and a chromatid, and then go through a little general discussion of how those numbers fluctuate during the different phases of cell division and why it's different during mitosis and meiosis. So first of all, a chromosome usually is defined as one single body, and what I mean by that is one chromosome can consist of one chromatid, and often that would be drawn sort of like that, or one chromosome can consist of two sister chromatids, often drawn like that, sort of looks like an X. Now, if we want to think about dolphins, since that's what they're asking us about, let's think about, first of all, mitosis, and think about the fact that a normal, I don't know if normal is the correct word, but in other words, a cell that's, let's say a cell that's in G0 of the cell cycle, that's about to go into mitosis. In other words, it's going to go through S phase and go through all of the parts and get to mitosis. I think I'll do it this way and say, if you were to look at just one of the 44 chromosomes in that dolphin cell, at G0, it would look like that. It hasn't been duplicated, so that each chromosome consists of one chromatid at that time, and therefore altogether there would be 44 chromosomes, and the number of chromatids would be exactly the same, right? And maybe while we do this, we'll end up getting some of our answers. As G0 goes to S phase, right? Or maybe I think I'll do it this way. Let's say that after S phase, right? Cells are going to be now in, you know what I just realized? I apologize for this. Cells that are in G0 would not be going on to divide, so what I meant over here is G1. I apologize about that, but it shouldn't really mess things up too much. So after S phase is complete, we're going to be in G2. I'm glad that made me remember that. And what really happens during S phase? Well, the chromosomes are duplicated, right? So if we looked at this same cell after it's gone through S phase, we're going to find that the chromosomes have doubled. They're duplicated, and now one chromosome consists of two sister chromatids, right? So we still have 44 chromosomes, but the total number of chromatids that exists at this time would be 88, right? Great. Okay, now we're going to go from G2, we're going to go into mitosis, right? And let's just say that after mitosis is totally complete, even though we're going to have two new cells that are genetically identical, if we were to look at one of them, the cells after mitosis would be back to having their chromosomes like that, because remember, the sister chromatids got pulled apart into two new cells, but in the one new cell we're back to 44 and 44. So that's sort of the general picture of mitosis. Now, think about a couple of things, though. It does depend what, if they're asking here, now let's go to what they're asking. They're asking about a specific phase of mitosis. Remember that anaphase is the time when the sister chromatids are being pulled apart. So they've already separated at that point. And that turns out to be important in getting the correct answer for this question. So perhaps I'll change ink color and go over here and ask ourselves, with respect to part A, in anaphase of mitosis, okay, we know that let's draw a cell, and we'll just again keep track of one of the chromosomes. Let's say this was metaphase of mitosis, right? So in this case, we have the doubled chromosome, well, lined up with the others, which are not being shown here on the cell equator. And at that point, right, we're still in this type of a situation. If we were to see all of the chromosomes, we'd have 44 of them, and we'd have a total of 88 chromatids, right? Now think about what starts to happen in anaphase. Now the two sister chromatids are being pulled apart from one another. But the important thing is they have separated. So we have to count each of these as a new chromosome, a single chromosome, which consists now of one chromatid. So here's what's sort of interesting. The same number of chromatids have to exist in this cell as they did before, right? And that number is 88. But how many chromosomes exist in this cell? Well, the thing is, as soon as these sister chromatids separate, you may not like this terminology. I'm not thrilled with it, quite frankly. They are now called each a new chromosome. So during the period after my metaphase and through anaphase until telophase is done, we'd have to say that there are 88 chromosomes in that cell still, and they're each composed of one chromatid. So the answer here would be during anaphase of mitosis, we have 88 chromosomes, each of which is one chromatid, so there are 88 chromatids there. Now, if this, I'm hoping it makes sense to you, but then let's take this to its logical conclusion. After telophase is done, these both become new cells, right? And at that point, we're going to see that each chromosome that exists in that cell is made of one chromatid, but it's going to be half the number, right? Because those chromosomes have been partitioned out to the new cells. So they didn't ask us this, but if we said, this is what we said here, if we were looking at this one of these two new cells, we would have 44 and 44 there. Okay, so I think I'm going to erase this part about mitosis, or at least part of it, and then we can see about what's a little bit different in meiosis. I'm leaving part because really the initial cell, whether a cell's going to end up going through mitosis or meiosis or not even do anything, the initial cell is the same, right? In this dolphin, it has 44. Oops, I erased something I didn't even want to erase. Sorry about that. What I was going to do is say this initial cell is always going to stay the same, right? Now, even if this is going to go into meiosis, it still goes through S phase and G2, right? So prior to meiosis, we still have this situation with 44 chromosomes and 88 total chromatids. But now let's modify this picture a little bit. Let's say after meiosis 1, what happens? Well, we'll draw our little cell. And now here we can't just write one chromosome because remember, the homologous pairs recognize one another and line up during meiosis 1. So we'd have a situation like this, and maybe in a different color, we'll write this one to show that they're similar chromosomes. They're members of a homologous pair. So in the case of the dolphin, just to make sure, I probably should have said this at the beginning, it said that this is a diploid organism where 2n equals 44. The haploid number would be 22, right? And the reason I bring that up is because that means that it's not relevant for mitosis, but that there are 22 pairs of chromosomes, right? So here we have one of the pairs. Now, in this cell, let's say again that this is metaphase of meiosis 1. In this cell right now, there are going to be a grand total of 44 chromosomes, right? And 88 chromatids. It's still in this configuration. But here's what's weird about meiosis 1. What separates during meiosis 1? Well, the sister chromatids do not get pulled apart, right? So even if we're talking about anaphase of meiosis, anaphase 1 that is, even though these are going to start to go apart from each other, we still are not going to be changing the terminology of the chromosomes. So until let's think of it this way. After meiosis 1 is done, right, then we're going to have, actually, because we're going to have in each new cell, out of the pairs, see, this gets a little bit messed up because we didn't consider the pairs before, but we're still going to have one member of the two pairs. So after meiosis 1 was done, we still have doubled chromosomes, but we're going to only have 22 of them, and a total of 44 chromatids. But so the answer, though, if we're asked about anaphase 1 of meiosis, or really any stage of meiosis 1, we're going to end up with this same number here, which is 44 chromosomes, 88 chromatids. Okay? But then they're going to ask us about meiosis 2, so let's keep going. And we'll say, what happens during meiosis 2? Well, let's start with a cell that now we know that this is one member of a homologous pair. We just said right here that this cell has 22 chromosomes in it, and there are 44 total chromatids because each chromosome consists of one chromatid. But now, I don't know if you've heard it said, but it's true that meiosis 2 is much more mechanically like mitosis. So sister chromatids get separated in meiosis 2, and as soon as they do get separated, we're going to have a situation where we have, sorry, you guys figured out that I'm not the best artist in the world, I guess. Where now each of these new entities, since they're separated, has got to be called a chromosome, right? We got the same number of chromatids in here until the cell's totally, finished with meiosis 2. So now we've actually, look, we started with 22 chromosomes, 44 chromatids. Wouldn't you agree that the same number of chromatids has to be there? It's got to be true. But now each of those chromatids, if you want, is actually one chromosome. So at this point, we're going to have 44 chromosomes. And since each of those chromosomes is made up of one chromatid, there would be 44. And just to finish this off, after meiosis 2, after this cell totally separated, right, now we're going to be down to 22 chromosomes, but only 22 chromatids, since they didn't ask us that one. I'm just trying to sort of finish the logic of it. 22 chromosomes, each of which is composed of one chromatid. So all this discussion, this is taking quite a while, but I think it's going to be easier to do the rest of the problems having done this. So with A, B, and C, they all asked us about anaphases of different things, of mitosis, meiosis 1, and meiosis 2. And we got our answers there. Now let's move down a little, and maybe we won't have to draw quite as much, because we're going to have D, E, and F. They're asking us about G1 and G2 in the mitosis cell cycle, and also prophase 1 of meiosis. I really think that we can use a lot of the information that we already have here, because when they say mitosis cell cycle, all they mean is the cell cycle in a cell that's going to go into mitosis. To be honest, it really doesn't matter whether it's going to go into mitosis or meiosis, because we've already got the answer here, don't we? We said that a cell that's in G1 is in this situation, and prior to S phase, there are exactly 44 chromosomes, each one of which is made up of a single chromatid. So we already did the work. Look, see, that was good that we did all that work. to get the answer that's sort of simple, I hope, 44 and 44 here. Now, just be a little bit careful. Sometimes there will be different configurations if you see similar answers. They're only asking us for the total number. They're not necessarily asking us for how they're moving or if they're sitting around doing nothing, so don't worry about that part. Now in G2 of the mitosis cell cycle, didn't we already say that? And we said the main difference is that G2 is after S phase. Things have already been doubled or replicated, so we now have the answer again. We still have 44 chromosomes, but now there's a total of 88 chromatids because they've been doubled. Now, what about prophase 1 of meiosis? Or another way of saying that is prophase of meiosis 1. You really could say it either way. Let's just say, even though this says mitosis cell cycle, remember we said even if it was going to go into meiosis, which we ended up leaving in our drawing over here, the first two, the beginning is going to be the same. So we said in prophase of meiosis 1, before anything separates, we're really going to be in the same situation as we were here. Because after G2, in prophase of meiosis 1, what's really happening is just that the chromosomes are condensing. They're starting to interact with the spindle fibers, but they haven't moved apart. So whether it was really honestly prophase 1 of meiosis or of mitosis, you'd still have to have the same number. So it's still 44 and 88 there. So far, so good. Now we're ready to go down and look at the things that you didn't see before, G, H, I, and J. And even though I just extended the page a little, it might behoove us to keep our little diagram in view for a moment. This part is not so bad, so I hope you'll agree. It says determine the number of chromosomes and chromatids present in the following dolphin cell types. Yep, that's what we've been doing. So a spermatid, a sperm, an oogonial cell prior to S phase, and a second polar body. So now these are really directed at gametogenesis, and they certainly have to do with meiosis. But we've got generally a pretty good handle on what happens, so what we really need to know is what the terminology is, okay? So in males, we can quickly say that the starting cell, that's 2N, is called a primary spermatocyte. After meiosis 1, we end up with, well, there are two of them, but we end up with a haploid cell that is a secondary, I won't write it again, spermatocyte. And finally, after meiosis 2, we end up with 4, I'm only drawing one of these, but 4 spermatids. Spermatids are genetically identical to sperm. It's just that the sperm that we all know and love has to mature. That's supposed to be an N in there. Has to mature. So my point is that by asking for the number of spermatids, the number of chromosomes and chromatids present in the spermatid, you might as well be asking about the sperm. But the spermatid is completely finished after meiosis 2, and we've already talked about that. That was the situation. See, it was good to be complete up here. Once it's all done and a gamete for all purposes has been created in this dolphin, we're going to have 22 chromosomes, each of which consists of a single chromatid, so it's 22 and 22. And then look, in H, it's the same thing. It's a dolphin sperm. So we just got finished saying that while a sperm is more mature in terms of the development of the cell, genetically it's identical. So we don't have to do any more work to figure that one out. Now, in female dolphins, or human women for that matter, you have a very similar situation, but instead of calling these cells spermatocytes and spermatids, we have primary oocytes, secondary oocytes, and the finished product is an ovum, since they're, well, it's an ovum, but there are differences. So in order to answer this question, we may not have to go through all of them. An oogonial cell prior to S phase just for the record, that means if it's prior to S phase, it hasn't even gone through. It's still, in our drawing, it's still over right at the beginning here, right? So remember, that didn't really even matter if it's going to go through mitosis or meiosis, but it's the starting cell. When they say oogonial cell prior to S phase, it's a primary oocyte. So it should be the same as this right here, and it is. So there's 44 chromosomes, 44 chromatids. The second polar body is the most difficult of this, but not because it's difficult. It's because you just have to know what a second polar body is. So the difference mainly between the mechanics of meiosis, I should say of gametogenesis in females and males, is that you have your primary oocyte, and after meiosis 1, you end up with two cells that are genetically identical, but this one is the secondary oocyte. This one is a polar body, which is not going to divide any further. The second polar body happens after meiosis 2 is complete, and you get haploid cells, but they have their sister chromatids pulled apart. So the bottom line is that a second polar body is genetically equivalent to an ovum, and in terms of chromosome number, that's exactly the same as a sperm, which we've already figured out, right? So it's a little bit sneaky. The second polar body, even though it never ends up doing anything, it's got chromosomes and chromatids in it, and it's genetically identical to the egg or the sperm, even though they wouldn't be in the same organism. So a second polar body would also be 22 chromosomes and 22 chromatids. And that, my friends, is rather lengthy, but it is the correct answers to these questions. And even though at the very beginning, guys, I said, let's just think about this, let's not worry about what they're asking right away, don't get me wrong. I mean, the biggest reason for people getting questions wrong, I think, or at least a big reason, is not because you don't understand the processes, it's that you fail to figure out what it is exactly that they want. I'll be honest with you guys, and this is just an interesting point, I did this before, and I got a couple of them wrong because I found out that I was thinking of it in the wrong way. Obviously, I understand the process, but it can get confusing, I'm just saying. So, you know, once we figured all of this out, then we were in a position to say, what are they really asking? So, yes, you do have to make sure you know what they're really asking. I'm sorry to keep moving this up and down, except I just want, there's no one view here. We can see where all of the answers are. So for A, B, and C, we've got that. For D, E, and F, we've got that. And for G, H, I, J, we've got that. All right, guys, be careful out there in the genetic world. I'll talk to you soon. Question 5. Edwards syndrome arises from trisomy of chromosome 18. A male with Edwards syndrome wants to have a child with an unaffected female. What's the probability that their child will have this syndrome? So how do we go about this? Basically, you guys know that in a, quote, normal situation, every chromosome number consists of a pair, right? And since we're talking about having a child, we're talking about meiosis and making gametes. So the question really is this. Under a normal situation, you have two homologous chromosomes, right? They separate from one another during meiosis 1. Then you have cells like this. And then meiosis 2, the sister chromatids come apart, and you end up with 4 cells, each of which should have only one member of the pair, right? Now, if you had Edwards syndrome, instead of having 2 trying to line up with each other, you'd have all 3 trying to line up with each other, and this is a problem for the cell. Almost always one of them will go one way and one of them will go the other, but what of the third one? So it either ends up going this way or it ends up going that way, and what that means is that basically after you were done with meiosis 1, you can't tell which one would be which, but you'd have one cell that had the proper arrangement, but you'd have another that had an extra. And when these went on ahead through meiosis II, these two gametes that got created would be normal, but each of these, I hope you could see why, is going to contain, can't draw a circle, is going to contain 2 and 2. So since he's having a child with a normal female, her gametes are going to be fine. So it basically means you can restate this question in this way. 50% of the gametes will give rise to a normal zygote, but this 50% of the gametes will give rise to another trisomic, right? Because the normal woman's egg is going to have 1 copy. So you're going to get 3. And if we go back up and we look at what the question was asking, the probability that their child will have the syndrome is 1/2. Because if you look at it this way, 2 of the 4 gametes that are produced will be, quote, normal. 2 of them will be aberrant in that they already have 2 copies. So 50% is the probability that their child will have the syndrome. Question 6 asks, how many human sperm cells develop from 25 primary spermatocytes in A, B, 25 secondary spermatocytes, and C, 25 spermatids? This question is easy if you understand it, but I guess that's true of everything. But we basically have to understand generally about gametogenesis in males is that you start off with the primary spermatocyte, right? And that primary spermatocyte is 2N. After meiosis I occurs, you end up with 2 secondary spermatocytes, and each of those now is already N, even though the chromosomes are still duplicated. And then after meiosis II, you end up with 4 spermatids, each of which is N. And a spermatid is genetically identical to a sperm cell, so just to make it, I can't draw very well, you guys know that, it looks like a lollipop, but that's supposed to be a sperm. Each of those is haploid as well. So once we have this picture, we can answer the entire question. She asks, I say she because Demetra wrote this question, but 25 is sort of arbitrary here. The bottom, you have to answer it, but here's my point. From 1 primary spermatocyte, how many sperm do you get? You get 4, right? So from 25, you're going to get 100. From a secondary spermatocyte, from one of these, how many sperm do you get? You get 2 from 1. So if you have 25, you're going to get 50. And lastly, how many sperm do you get from 1 spermatid? You get 1. It just develops into a sperm. So if you have 25 spermatids, you're going to get 25 sperm. It's really as simple as that. Question 7 asks, how many human egg cells develop from A, 25 primary oocytes, B, 25 secondary oocytes, and C, 25 polar bodies? So if you did question 6, this is very similar, but it's trying to highlight the differences between female gametogenesis and male. So we'll draw a similar diagram that says in females you start out with a primary oocyte, which is diploid. Now after meiosis I, you end up with 2 new cells that are haploid, but here's where the difference begins. One of these is a secondary oocyte, and I know they look identical because genetically they are, but the secondary oocyte in fact would be a lot larger. This is a polar body up here. That polar body is going to lead to nothing. But this secondary oocyte will go through meiosis II, and it will make an ovum or an egg cell, which is what the question asks. An ovum equals an egg cell, right? It will make one and then another polar body. So again, if we understand this relationship, we have 25 here, but we want to ask ourselves, how many mature eggs come from one primary oocyte? And the answer is only one, right? So if there are 25 primary oocytes, you're going to get 25 eggs. How many eggs, mature eggs, come from a secondary oocyte? Well, again, only one, because you also get a polar body. So you're going to get 25 secondary oocytes, 25 eggs. And C is sort of a trick question. 25 polar bodies, well, you could have a million polar bodies. The point being, polar bodies do not give rise to eggs. They're arrested, frozen, and they do nothing. So how many eggs do you get from 25 polar bodies? 0. See you next time.